self and non-self that allow them to recognize structures or recognize molecules that are conserved amongst different groups of microorganisms that are not present on host cells. And therefore macrophages can recognize non-self. So when this happens, these toll-like receptors that recognize microbial components, that initiates, that signal is transduced to the nucleus. So a signal transduction event takes place that upregulates, that will be responsible for the activation of a very common transcription factor in immune cells, which is NF-kappa B. And so the release of NF-kappa B from its inhibitor will allow it to translocate to the nucleus and transactivate genes that encode cytokines and chemokines. So what are cytokines and chemokines? Cytokines are essentially hormones of the immune system. And these are hormones of the immune system are secreted by macrophages. So there's an initial recognition event. Macrophages have detected a foreign invader. These are sensors for pathogens. That means macrophages, they have a seventh sense. They can see danger that we don't see. And so once they sense a pathogen through these receptors that have broad specificity for the various constituents of microorganisms, this induces the transcription of cytokine genes and chemokine genes. And cytokines function as hormones of the immune system in that they will control responses. They will bind to high-affinity receptors on recipient cells or neighboring cells and bring about changes in the biology of those cells. On the other hand, chemokines are also secreted, and chemokines are chemoattractant type cytokines that will stimulate the specific migration of cells. So let's go back to this slide. The physiological changes that are taking place in injured vascularized tissue where a microbial pathogen has been introduced in this scenario. So how do we make it physically possible? How do we deliver defense mechanisms onto this site of invasion to detect the foreign invader and remove the invader before an infection becomes established? Macrophage cytokines that are produced in response to sensing bacteria will bind to specific receptors on capillary endothelial cells and bring about changes in the biology and structure of these cells, such that the capillaries will become leaky and will become engorged with blood. And that makes it possible for plasma proteins to come in and react on the surface of the bacteria to make recognition and elimination much more efficient. But on the other hand, chemokine proteins, or chemoattractant type proteins, establishes a chemical trail that neutrophils and other white blood cells in the bloodstream can pick up, can sense this chemical trail through their chemokine-specific receptors and migrate to its source. So they're going to follow a chemical trail from the capillary endothelial cells and follow that trail to its source. By that time, all of its chemokine receptors will be most likely saturated. So macrophages are attracting cells into the site. So when neutrophils are attracted to the source of that chemokine, they're also in the area of the microbial invader. And neutrophils, like macrophages, can sense the microbial invader. And their job is to engulf the bacterium and kill the bacterium inside of phagocytic vacuoles. And that's illustrated here. Macrophages also have another function. In addition, between standing guard and sensing a breach in one of the gates and distinguishing between self and non-self, and then focusing defense mechanisms on that site if there is a breach, macrophages also participate in elimination of the pathogen itself in pretty much the same way neutrophils do. They can engulf a pathogen. They also have broadly specific receptors that bind to surface constituents of pathogens, which will induce a change in cytoskeletal organization to allow them to engulf a microorganism into a phagosome that will merge with a lysosome containing lytic enzymes that will degrade and kill the microorganism. And that's essentially the job of the neutrophils. They're very actively phagocytic and bactericidal. And so those are the recruits or the backup that macrophages attract, bring into the area once the body's been invaded. Okay? Are there any questions so far? If you have a quick, yes, ma'am.
Would you equate the chemotaxis of the neutrophils or the phagocytes to quorum sensing with bacteria, how they sense the gradient and they move toward it if it's a chemoattractant?
I'm not sure. That doesn't. Oh, you mean does the chemokine affect the bacteria anyway?
No, I mean the phagocytic cells that are trying to find the bacteria.
Mm-hmm.
There's the chemokines that they're trying to go toward the concentration gradient where it's more concentrated.
Mm-hmm.
That's how bacteria sense, say there's food or nutrients, they want to go toward it. Is it a similar mechanism that they use?
I believe so. So quorum sensing allows these microorganisms, let's see, in a favorable environment, they start to proliferate. In an unfavorable environment, they probably wouldn't, to avoid being eliminated by the immune system. But once they find a favorable environment, then they can expand their numbers. I believe, if I understand quorum sensing right. And neutrophils can sense their environment in pretty much the same way. They have specific receptors that recognize cytokines and chemokines. And so the chemokine essentially would induce the directional migration. So as the chemokine is secreted by the macrophage, so it's going to be bound in the interstitium here. On these collagen proteins and some of these glycoproteins, it'll remain bound to increase its half-life. And so neutrophils, as they enter, they're going to express cell adhesion proteins that will allow them to crawl through this matrix of glycoproteins. And as they sense these chemokines, that's going to induce them to follow this concentration gradient or trail. Did that answer your
Yes,
question?
it did.
Okay. Are there any other questions? So the take-home message here is that macrophages orchestrate an innate immune response once they recognize the tissue damage along with a foreign invader. And most likely, tissue damage will always involve the introduction of a microorganism. And so it's the job of a macrophage at that point to focus defense mechanisms onto the site of invasion. So these are basic defense mechanisms that are designed to eliminate any type of microorganism that could challenge the body and prevent the establishment of an infection. So this is it's called non-specific immunity because it's non-specific as far as the type of pathogen is concerned. It reacts the same way to remove any of the different classes of pathogens that could enter the tissues to prevent an infection. Okay, so let's move the scenario just a little bit further and say the microorganism has been able to overwhelm the innate defenses and establish a site of replication or establish an infection. And once that happens, our third layer of protection, so the first layer are the epithelial barriers that protect the healthy tissues from our microbial environment. The second layer actually constitutes the immune system, the innate immune system, which will respond if those protective barriers are breached. And then finally, the third layer, which constitutes the immune system. So there's actually two layers of immunity, innate immune system and adaptive immune system, that are only activated once the primary barrier is breached. Most likely, in most cases, the innate immune system is able to eliminate a foreign invader and prevent a pathogen replication from becoming established, and you do not come down with any signs and symptoms of sickness, and you never know that a foreign invader was ever present. But in other cases, microorganisms have evolved ways around the innate immune response because they're trying to be an ideal parasite. Once they enter healthy tissues where there's plenty of nutrients, the appropriate pH and the appropriate temperature, they try to be the ideal parasite and avoid being eliminated. And it's some of their characteristics that they've obtained over time because prokaryotes are replicating faster than eukaryotes. The problem is some of their virulence factors or disease-causing power that they use to acquire an appropriate environment, such as healthy tissue, can also interfere with vital functions of organs and cause disease. And so, in this particular case, the innate immune system, once it is overwhelmed, then an infection becomes established. Microorganisms, as well as their toxins, can therefore enter the lymphatics or the bloodstream and interfere with vital functions of organs. by this time you start to become sick. When that happens, when the innate immune system is overwhelmed, then the adaptive immune system is activated. So why would the innate immune system be overwhelmed in the first place? Well, that's really due to, there's a quantitative reason and a qualitative type reason. The quantitative reason would be sheer numbers of microorganisms that might be introduced into a cut that are rapidly proliferating microorganisms that it's going to take a couple of hours to bring neutrophils into the area. So depending on the number of microorganisms and their rate of replication or cell division that enter a cut, they can establish a focus of infection quite readily. And at that point, it's just the goal of the innate immune system to try to hold the infection at bay while the adaptive immune system becomes established. And then there's the qualitative reasons. Some pathogens have evolved ways around being eliminated by the innate immune system. So there's some pathogens that can antagonize phagocytosis, and some pathogens that can antagonize being killed inside of macrophages and neutrophils once they've been engulfed or phagocytized. And therefore, that makes the elimination of the microorganism very inefficient. And so that allows the prokaryote to get the upper hand and establish a focus of infection. At this point, that's when the adaptive immune system is activated. The adaptive immune system is activated. This particular type of immunity is specific for the pathogen itself. This results in specific recognition. So it's not non-specific type recognition or broad specific type recognition that the innate immune system uses. The innate immune system, its goal is to prevent an infection from becoming established in the first place. Therefore, it's got to sense that all microorganisms in the environment. The adaptive immune system, once the innate immune system has been overwhelmed and infections become established, and we start to see signs and symptoms of the disease, we start to get sick at this point, then an immune response that is dedicated to the pathogen itself is required to eliminate that particular pathogen and deal with the characteristics of that pathogen that allowed it to overwhelm the innate immune system in the first place. And since this is obviously a dangerous microorganism that will be endemic to the environment of this animal will encounter this pathogen again. And therefore, another key of the adaptive immune system is that it will remember the pathogen, such that if the animal encounters the pathogen again, the immune response will be much more rapid and efficient to prevent an infection from becoming established a second time around. Or at least if an infection does become established, it will prevent disease. It will prevent you from having the same disease the second time around. So how is the adaptive immune system activated? There are other cells that are present in the tissues along with macrophages, abbreviated here DC or dendritic cell. And that's because of their morphology. They have a starfish-shaped type morphology when you see these cells in tissue sections. And essentially, the important thing to remember is these DCs or dendritic cells are antigen-presenting cells. They're responsible for activation of the adaptive immune system. Macrophages are responsible for activation of the innate immune system. If the innate immune system is overwhelmed and an infection becomes established, then another class of white blood cells or leukocytes, dendritic cells or antigen-presenting type cells, are responsible for the activation of the adaptive immune system. And so the antigen-presenting cells will engulf the pathogen that has established a focus of infection and then migrate to specialized tissues and make antigens derived from that pathogen available for the class of white